I will take you like 15 to 20 minutes just to share what my recent work, and also previous work. So this is quite recent. I actually presented it in Bangkok last 3 weeks. So the project is about the coral, the effects of anoxia and hypoxia on sclerotinia corals oxidative stress responses. So this project is actually running in Thailand, but part of the oxidative stress, we bring the sample back to our lab here. But the experimental we did in Thai University. So this is pretty much me. So what I did, my field is marine biochemistry. Some of you asking what is actually you doing, but I've been around here and there. So I'm a biochemist, marine biochemist in background. So my research interests more on environmental and redox proteomics. Oxidative stress biomarkers, pretty much same like Mark doing on that side. And also part of marine biotechnology work as well. So I'm under research interest group. So in UMT, we have few research interest group that we call RIG group. So I'm under research and education on environmental for future sustainability, short form as REEFs. Our group pretty much do everything related to marine environment. So this is our group, familiar face. James, Hafiz, there's Po, Safwan, and our mentor, Professor Zainuddin. So our core is actually working more on coral reef research. James might give this as well to you about restoration work that we've been doing in Bidong. So that's actually our work mainly. So from the new project, we have project working with Rigs to Reef project, and also another restoration work in new site, which is in Kapas. So now we have 2 sites of restoration, which is in Bidong. The main purpose of Bidong is to increase the population of coral in that area because of Pabuk storm. But Kapas, new site, is actually engaging with the local communities in Kapas on restoration work. So the book that you have is part of output from our project, and there will be a new output of restoration book in near future. So this is generally what I've been doing in my lab, redox proteomics for environmental monitoring. So pretty much I'm working with a few marine organisms, mainly coral reef, and also bivalve, which is oyster. Oysters, mussels, meretrix, the one that you collect today, is part of my research animals as well. But that one is more on toxicology study and also stress study, of course. All right, so most of the methods is doing enzyme as the biomarkers, and after that, looking at the changes of the protein as a biomarker. Well, I might skip. This is the principle of oxidative stress, lack of oxygen paradox, because I mentioned the topic is hypoxia and anoxia, so it's involved more on oxygen. So this one is just, I want to explain previous years, like 4,500 million years ago, the Earth is actually limited of oxygens, and after maturity of the Earth, and then it's become oxidized environment, and involvement of oxygen is more. So pretty much living organism, we cannot live without oxygen. So the statement, the oxygen is hard to live with, but even harder to live without. If we have more oxygen, it's also toxic for us. So there's a certain level of oxygen needed for living organism to make it par condition, like normal condition. Well, this is what we discussed during the conference about the oxygen paradox in seawaters. If the high temperature happen and oxygen level also drop, at the same time. So maybe the clear example is from this next table. We call it like extensive hypoxia and anoxia and a minimal hypoxia and anoxia. The scientists can predict certain of the area, which is if they have high temperature, the prediction hypoxia in the line of that after the high temperature because of the high nutrient. Okay, especially on the phytoplankton, for example, they will bloom so fast after the high temperature occur. So in Terengganu coast, we don't have condition hypoxia. But in Thailand, quite few patches, if they high temperature, the nutrient, which is the algae, will bloom so fast, and that area become more murky, so it's lacking of oxygen. So from this project in the field, my collaborator Martini, she actually taking sample from both high hypoxia level and also some place that have totally no oxygen, which is anoxia, and then they transfer into the normal place to see the recovery, which is, that's a recovery sign of it. But on this experiment, we actually collecting the sample from the hypoxia area and bring to the lab and see how fast it can give harm. So I always using this image to give understandable about oxidative stress. Anything about oxidative stress, we will relate to us first. So if we are not well, what happened in our body? Our antibody start to increase. And at the same time, the level of oxidative stress also increase. So antibody helping out of this oxidative stress level to balance it out. So it's happened also in marine organism, in other animals in the environment. So in coral, it's quite dynamic because it has symbiodinase with them. So the big question is that which one gets stressed first? Is it the symbiodinase or the coral tissue? So it's very complicated to answer the questions. So to avoid, because it's a primarily study as well, we didn't separate the symbiodinium with the coral tissue. We mix it together. So we want to see the total amount of protein in both symbiodinase and also the coral tissue. And from there, we can measure the enzyme. So this is the principle behind the oxidative stress, which is the ROS, reactive oxygen species. You can see the example of different reactive oxygen species, which is superoxide, hydroxyl, peroxyl, and so on. So it depending on what type of oxidative stress enzyme that you want to use in your experiment. And this is a very good example of model. It's actually from this publication. We use this as a reference as well to see the mechanism happen in the coral from the photosystem 1 and photosystem 2. And all of this involving the SOD, for example, part of the defense, and catalase, and also glutathione S transferase. And this is the simplified principle of it. Okay. So this is the area where we collected the sample in Kam Island. Is a southern part of Thailand, in the Gulf of Thailand. So this area is quite unique. It have very fluctual temperature as well. And you see the data have been collected from August 2021 until October. So in between that time, you can see the dissolved oxygen is below the normal, below the 2.0 milligram. So we want to find out what happened is in the laboratory scales, what happened to these different species of coral. So we selected 3 different species of coral, which is Poecilophora acuta, Poraitis lutea, and Turbonaria mesenterina, to see which one have faster effects after we introduce with lack of oxygen. So the exposure time is maximum 72 hours. Okay, so you can see from this experimental design. And this is standard protocol when the experiment, you collect the sample and then you use airbrush to take out of the tissue and also the symbiodinase, and then we homogenize it, we crush and then centrifuge and we do protein concentration assay and proceed for the enzymatic. So for the coral tissue condition and symbiodynamic density, the most affected species with a sign of rapid tissue loss in Fossiliifera acuta. So the mortality that we see from Fossiliifera acuta in anoxia treatment was observed with tissue loss in replicates within 24 hours. So from our first observation, we already know, okay, Fossiliifera is the most vulnerable species out of 3 species that we collect. So Poraitis lutea and Tubernaria mesenterina showed no changes in tissue condition within 72 hours time frame. So looking at the experience from previous temperature exposure from previous study, we know that some of these species, especially Poraitis, have quite resilience compared than branching coral. So it's not give us surprise at all on this experiment. And on the oxidative stress enzyme responses, you can see the simplified here, anoxia and hypoxia. The catalysts have higher for Fossiliifera acuta, which is, I said at the beginning, that's not surprise at all. And then it also gives, this species also showed the same effects with SOD and GST. And the good news from this experiment, we know that Poraitis have very strong and resilience. So knowing from this, we believe that the Poraitis species will be the next species that we focus for restoration work. Like branching coral is good. It's good to go for fast growth coral for restoration, but Poraitis have very slow. So it's tally with the normal condition of the coral reef ecosystem. The hard coral, the Poraitis, for example, not the branching, the stony coral have very slow grow. So it gives chance like for the branching to grow more. But the vulnerable for the branching coral is very sensitive. So it depending on the condition during that time when you want to select which species for the restoration. So a series of antioxidant enzymes whose role is to intercept and inactive reactive oxygen, intermediate and synthesize all known aerobic organisms. So this is explanation behind on the biochemistry is that although extremely important, the antioxidant enzyme and compound are not completely effective in preventing oxidative damage. It also depending on the condition itself as well. So to deal with the damage that does still occur, a series of damage removals, repair. So although we're looking at the experiment, for the example, we do temperature exposure. After the high temperature in certain time, we leave it in the aquarium for a certain time with very good condition. There's a chance for this coral to recover again. It's not totally bleach. Sometimes when you look at the microscope, there's a sign of evidence of the healthy tissue as well. So there's a chance for it to grow. All right, for this experiment, the concluding remark is that the present findings suggest the Poraitis lutea is resilient to some extent to short-term exposure to anoxia and hypoxia. Bear in mind, it's very short-term exposure. Maximum is 72 hours. It's probably give, we don't know, in 1 week exposure, it might be different results, but in short time, we know that Poraitis lutea have that resilience. So these effects are unique indicator for coral resilience, emphasizing the importance of studying oxidative stress response to better comprehend the resilience and adaptability of the corals in the face of changing environment condition. And such insights are paramount for the conservation and management of this invaluable marine ecosystem. So it's actually the project involved all these people together. Martini, Longying, Arif, and also Verdiana. So part of that, we also have another project going on the temperature. So I skipped, the principle is pretty much the same. We collect the sample in the field. So this is another technique that we use, part of enzymatic, which is we use protein labeling, IAF labeling. This one is what we call it protein trap, using the fluorescent trap to understand which protein is response towards that stress condition. And after that, we go for gel electrophoresis, running the gels in the lab like you do genomic study, but this one is vertical method. Okay, just skip this one. So from, just give you an overview of output using the fluorescence tagging is that when we do the separation of the protein, we use another scanner, and the scanner will pick up the fluorescence labeling that you will compare with the stain that you use, pretty much looking like this. So using the software, we will see which one have higher intensity, and then we will focus which molecular weight of the protein, and then we cut it and then trypsinize and send for identification and to see what exactly protein that responds during the stress time. So this is the technique if you want to do next two-dimensional electrophoresis, but in the lab, at the moment, I cut this method because I already jumped to shotgun proteomics, which is I skip this. Shotgun proteomics is directly after trypsinization we send to the company, but it's a bit pricey compared than this method. But this method is time consuming. Yeah, we already did this in the lab. So for ARIF project, we managed to identify I think 2,300 protein responsible for stress. After that, we need to segregate according to their functional. So these three people already graduated. Safwan become our academic staff now, is part of the project at the moment with coral restoration. Arif already graduated his PhD. Atika already graduated her master, now working in, I think in one government science agencies. And this is my collaborators from Thailand, Martini and her student. The project also on the Maiton Island and Panwa Island in Thailand. This place have very high turbidity, but there's a coral survive in that area. He's also graduated already, now currently doing PhD in different institute. He measuring oxidative stress regulation in lattice calcarifer, which is part of it in here, collected from Setiu farm on ikan siakap, lattice calcarifer. I have a bit of a project on sea urchin larvae development, also looking at oxidative stress enzyme as well. And this one, the one that you collected, so this is meretrix we collect from Setiu, but this part I just focus on the hemolymph, which is the blood of the meretrix. What color of meretrix blood? That's horseshoe crab blood. What color of meretrix blood? Hemolymph, we call it hemolymph. Transparent. It's not supposed you to answer. It's supposed to be them. Yeah, it's transparent. But from this hemolymph, like in oyster as well, it's transparent. But from oxidative stress study and hemolymph, it's really good to understand the impacts of the environmental stresses in that area. Yeah.
Where do you take the hemolymph from? Do you take it from the adductor muscle?
Yes. Yeah. Same method like we use with oyster. And I also have project with Bill as well on oysters here, and also oysters from Scotland as well. It's ongoing project. We receive a grant that, together with Mark, also in the committee as well. We call it IPRG grant, International Partnership Research Grant, which Nikki is the candidates for masters on that project, and part of it Alex PhD project. Yeah, so this is the team we've been working on since ages, that's what we always laugh, like we will grow old together. Because James and Po, we are same batch undergraduates until now. So technically 23 years now, been working together. Yes. Yeah, with that, thank you. Yes.
For the varietal restoration, is the process similar to how you do restoration for branching corals, or is the process quite different for varietal?
The process, can you detail what process you?
In terms of how you get the bits of coral that you
The process is the same.
It is the same.
Yeah, you put on top with the epoxy and all that is the same.
I was wondering why you picked those 3 species in the first study, the 3
Ah.
species of coral.
Okay, that one is the most majority species that can found in that area. Yeah, I think that's the first thing that we observe when you want to select the species. You will look at the most abundant species in that area.
Why does the oxygen oscillate so much in Thailand? You know, seems to be a lot.
Yeah, it's a lot. Part of it is temperature changes. If you see from August, it's quite high temperature, and it will drop close to the monsoon season because this is southern Thai. It's also have pretty much the same like us, like dry and monsoon. And the other thing is that part of the area quite muddy. It's very unique in Songkhla area. There are geographical. If you see the map, it's quite. It's a lot of discharge from the mainland as well.
But those oscillations seem to be nearly like daily or weekly, not.
Yes, it's a weekly.
Yes.
Yeah.
Why is that? Is that
I have no idea.
not just temperature like seasonal
Yeah.
temperature?
Yeah,
Day and night.
part of it is discharge from the mainland.
Yeah.
Mm.
And the other question was, what is the health of the corals there? I mean, they can be very healthy if the temperature is so low.
They survive. They survive part of the coral. They have also patches like bleaching as well at that time. Yeah.
Is hypoxia always associated with bleaching?
Yes.
So even if, let's say,
The
you've got-
bleaching happen regardless temperature or hypoxia. It's when the coral gets stressed,
Yeah.
regardless what stressor
so
around
hypoxia
them.
is not necessarily a driver for
Not necessary.
bleaching.
Mm. The sign of bleaching of the coral is the sign of stress. Yeah, discoloration first, and then when it become more worse, it's become bleach.
Do you know if there's any studies that have been done on the oxidative stress response to fragmentation in corals?
Oxidative stress and
To fragmentation, so if post-cyclonic high energy events that break a lot of corals. Do you know if there's been any studies done on the stress response to the breaking of
the break of the
Yeah.
coral? We have discussed on that, especially on Sabrina work, comparison with the restoration site and that. For the restoration site, we use the coral that break, but there's a certain time for the normal condition for the coral back when we do restoration. But when the coral been break, there's we will call it acute oxidative stress. But in the normal condition, when it gets stressed, this oxidative stress already in the system. When am I sentence right? We play with the acute stress, and also they're already in that particular stress condition in certain of time. So that's where my earlier explaining about the balance between the antioxidant and also the oxidative stress level. So that might spike. If you do experimental on oxidative stress, maybe in 1 or 2 days you can see spike on the oxidative stress, and then you see again how it become normal. It depends. there is possibility of another Symbiodinium to settle on the coral.
And so is it seawater you're using in your tanks that already has a zooxanthellae within it that then repropagates the coral?
No.
So where does the zooxanthellae come from to repropagate the
No,
coral?
no, I didn't do that in my experiment.
Oh,
There's a
sorry.
possibility from the reference
Okay.
mentioning.
Okay.
If in the tank, if in nature, you have the possibility of Symbiodinium come back and settle, come back in the coral itself. But in the tank experiment and in the future, I would say that you need to put the Symbiodinium. But some of the lab also culture the Symbiodinium. Yeah, they did a lot in culturing Symbiodinium.
So your choice of experimental corals is with a view to restoration, so I'm just thinking back to the 1980s when there used to be big cropper elkhorn forms in the Caribbean, and those have mostly died right back now. So I'm wondering whether there are other species that have already gone from the system or been reduced dramatically in these sort of areas. Has that happened over here? I don't know.
We don't have huge bleaching event here in Malaysia much. Patches, yes. I think James will be the right person to answer that question.
So is part of the end goal trying to isolate whether it's protein or some kind of genomic specificity to the chlorides and then try to transfer it so that the resistance goes to other corals, or is it just noted as being hazardous?
No.
Possible?
No. Some of the work I think do on clades. Arif is not here, I think. Symbiodiniaceae, we looking at the clades. Clades A, clades B, clades C, clades D. So some of the research from the reading, not from my experiment, they select the clades that have, for example, heat tolerance, and do on tank experiment first for that. But they did that. There's been research on resilience clades. Thank you. So, we also want to record Hamish as well.
We'll just take a 5-minute comfort break while we swap the speakers round.